शिखर हासिल किए हैं। वैसे हमारे प्रिंसिपल सर डॉक्टर अंकित बी चौधरी सर का मैं परिचय देना चाहूँगी। Dr. Ankit B. Chaudhary sir is currently working as Principal, Saraswati Institute of Pharmaceutical Sciences, Gandhinagar, Gujarat. Sir has completed his MPharm and PhD from Ganpat University. Sir has completed PGDRA in 2021 from Gujarat University. Sir has credited 6 books and many research and review articles in reputed journals in his credits. Sir has received more than 75 lakhs rupees grant from various government bodies for various research projects. Dr. Ankit B. Chaudhary sir is currently the member of APP Gujarat State. Sir is Vice President of SKPCER Alumni Association. Sir is currently Treasurer of APTI Gujarat State Branch, as well as Sir is the member of PCI, which is a very graceful moment for the SIPS family. Now, I would like to invite our principal, Sir Dr. Ankit B. Chaudhary, to deliver the welcome note to all the participants. Please, sir.
Hello, am I audible?
Yes, sir.
Okay, so thank you very much, Charmi madam, for giving my kind introduction. So I heartily welcome all the participants in our one-day seminar. So our institute, Saraswati Institute of Pharmaceutical Sciences, it was established in 2006, and we are running Bachelor of Pharmacy, Doctor of Pharmacy, and Master of Pharmacy. Master of Pharmacy in Quality Assurance and Pharmaceutics. Our institute is accredited by National Board of Accreditation last year only for BPharm course. So our institute is committed for quality education with faculty, resources, and very good infrastructure. So if we talk about today's national webinar, the topic of the webinar is validation and data integrity in pharmaceutical industry. So this kind of webinar we are arranging to minimize the gap between industry and academia. And all the three speakers we called from industry only. First speaker is Dr. Hareshkumar Patel. He is Senior Manager R&D, Sun Pharmaceutical Industry Limited, and his topic to deliver is computerized system validation in pharmaceutical industry. Our second speaker is Mr. Yogesh Mandalia. Yogesh Mandalia is our alumni only. He did MPharm from our institute, and he is currently working as a manager of audit and compliance, Sun Pharmaceutical Industry Limited. He is going to deliver a topic on industrial approach on cleaning validation. And third speaker is Ms. Fozia Qureshi. She is also our alumni. She did MPharm from our institute, and she is going to deliver a topic on importance of data integrity in pharmaceutical industries. She is working as a subject matter expert and corporate trainer at Vida Clinical Research. So I heartily welcome on behalf of our SIPS family to all three eminent speakers. If we talk about total participation, then in this webinar, total 787 all participants have participated in this webinar. And if we talk about the state throughout India, this is national webinar. So total 17 states have participated in webinar. Total students are 616. If we talk about assistant professor, then 104 faculties as assistant professor, they are going to join. Then associate professor 28, professor 20, and 7 principal, even 7 principal are also going to join the webinar and will get benefited. If we talk about industry person, then 8 persons are invited. So I heartily welcome all the participants throughout India from academia as well as industry to be present and for accepting our invitation. So I request Madam, to continue the webinar. Thank you, madam.
Thank you very much, sir. Now I would like to request to introduce our first speaker, Dr. Hareshkumar Patel. Sir has more than 18 years of academic, research and industrial experience. Sir has completed his B.Pharm, M.Pharm and PhD from LM College of Pharmacy, Gujarat. Sir has awarded fellowship from AICTE for the post-graduation. Sir is currently associated with Sun Pharma in the Department of Pharmacokinetics. Previously, sir was associated with Cadila Pharmaceuticals and as research executive and research scientist as Alembic Limited. Sir has more than 25 research articles in the very reputed peer-reviewed journals. Sir has delivered many presentations and conducted many trainings at various organizations. Sir has faced many international and national audits such as FDA, MHRA, NVISA, etc. Sir is currently associated with quality management system and its tools. So I would like to welcome our first speaker, Dr. Hareshkumar Patel, sir. Please, sir.
Yes, good morning, everyone. I'll just confirm that, am I audible?
Yes, sir.
Okay. Thank you for the confirmations. First, I would like to talk about Saraswati Institute of Pharmaceutical Science. And I am so thankful for giving me the opportunity to deliver the lecture in front of, I can say, more than 2,000 to more than 200 participants in this program. And I would also thanks to the organizing committee, especially Dr. Navneet sir, for the giving me the opportunity and for the give me a chance to deliver such a grateful seminar in front of grateful audience, because I can see, as Dr. Chaudhary sir already talked about the audience and the compositions of audience from the, I can say, from the students, professor, assistant professors, and including a principal. So I am sure that this audience can get the more benefits from this presentation. And with this word, I will just start my presentations. Now my screen is visible to all of you. Please confirm, anybody from audience?
Yes, sir.
Yeah, thank you so much, sir, for your confirmations. Again, welcome to all of you for the presentations on the topic is computerized system validations in pharmaceutical industries. And thank you, Charmi, for give me the nice and kind introductions from the college life to till current experience, considering current experience. Now let's, by the end of this presentations webinar, we are able to understand different regulations and guidelines applicable for the software validations, computerized systems, what are the concept and why it is required the software validations in pharmaceutical industries, the applications of V models for the software validations, what type of workflow we have followed during the software validations, who are the stakeholders involved for software validations, including from the technical, non-technical, I mean from the IT background, from the pharmaceutical background, from the quality assurance background. What are the different categorizations, or we can say classifications of software, and other important aspect regarding the software validations. And finally, we will see the one example as a demonstration, and at the end we will summarize the presentations in single slide, Hope you will get the benefits from these presentations, and from every slide we have a one take home message, so you can pick the message that can be a relevance to you. So before I'll start the presentations with the regulations and guidelines. So why it is a required why software validations is required in industry? If you see the ISPE GAMP 5 guidelines, I mean the Good Automated Manufacturing Practice guidance published by the International Society for Pharmaceutical Education. And in this guidance, there is a series of guidance is there, GAMP 1, 2, 3, 4, but the recent current one is a GAMP 5 guidance is there. And it is a talk about the every industry, every pharmaceutical industry. And if you are using the software either in your production environments, in research and developments, for the quality control laboratory, or in maybe for the quality training, or in pharmacovigilance, where you have to use the software for the reporting of adverse drug reaction. So wherever software is involved, we have to provide the validations data, because what happens if you use the software which are not invalidated in nature? So what we can conclude, is it possible to get the accurate and appropriate result which we expect from the software? So it's not that. And that's why the confirmations and validations of software is a mandatory to provide the human related data. And as we know, in pharmaceutical sectors, we are dealing with the medicines, pharmaceutical product, and which are directly taken by the patient, or we can say consumer risk is there. And considering that risk, software must be validated as per the guidance requirement. Now let's talk about the another guidance. So here it is clearly says the electronic record and electronic signature by the Food and Drug Administration. So this is the FDA guidelines. So whenever we talk about the export product, I mean especially for the US market, we have to satisfy the requirement for electronic records and electronic signature. Now the questions came in mind that, what do you mean by electronic record? So electronic records means whatever records during the analysis, during the manufacturing of product, directly capture into the software, and this data is used to support the safety, efficacy, and quality of data. So that is known as the electronic data. Now another part, we talk about the electronic signature. So what do you mean by electronic signature? Meanwhile, whenever we are performing any activity by the software, and we have to put the electronic signature, I mean unique user identifications and signature. Let's see the example of suppose if you want to use the net banking. So in net banking, what is there? There is a two term. One is the user ID, and second one is a password. So user ID is a unique. It's a individuals, and it's a give the authentic user. Only this unique ID is given to that authentic user. So that is the one part. Second part is the software. It's about the password. So again, password is, and why password is required? Because nobody can access your data, nobody can see your transaction, nobody can see transaction on behalf of you, and that's why user ID and password is a mandatory requirement. So this is the one type of electronic signatures or electronic authentic credential used for the banking. Likewise, in pharmaceutical industries, whenever we review, we are create the document. Suppose if you prepare the standard operating procedure, so what happens? The persons create the SOP. So they sign as the creator, or they can sign as an author. So that is the one type of electronic signature. Once the electronic signature done by the author, this also further review by the department side, or maybe from quality side, and finally it is approved by the head of department or head of quality assurance. So all signature will be considered as an electronic signature. And whenever we use the electronic signature, it is a mandatory to follow the ERAS requirement from the Food and Drug Administration, especially if you are working with the marketing your product into the export for the export market. And one more guidance, it is related to the Annex 11. And this is especially applicable for the European regulation, where it is clearly said that every software must be validated. However, if you see this slightly, there is a difference. stringent requirement mentions in Europe, and some may not be applicable in GAMP 5 or we can see ERES record guidance. But little bit, if you follow the stringent guidance for your software, you can satisfy the requirement for the all countries. And that's the reason we are performing the software validations by using a stringent criteria. Let's understand the history, or we can say how it's come in the regulations. So in 1991, GAMP 1, GAMP Good Automated Manufacturing Practice came in existence in first time, and it's introduced for the pharmaceutical applications. In 1992, Annex 2 is come in picture, and after that, in GAMP 2 further released in 1996 based on the whatever the requirement from the annex, and what is the comment received from the stakeholder from the different industries. After that, based on all three guidance, FDA also released the 21 CFR Part 11 1997 in 1997. So what do you mean by 21? 21 is dedicated for the drug product. What do you mean by CFR? CFR is a code federal regulation, especially the code is given specially for the US FDA. And Part 11 is specially denoted for the ERES record. And that released in 1997. After that, again, the guidance, it's come in notice that we have to revise the guidance GAMP 5 because there is a gap between the 21 CFR Part 11 and GAMP 2 guidance. So based on that, again, the GAMP 3 released in 1998. Again, based on the input from the industry perspective, it is also revised in 2000. It was also revised in 2001. And finally, based on all the evidences, FDA again concluded and revised the 21 CFR Part 11 in 2003. And after that, all together, and we can say current and effective version is a GAMP 5, which was released in 2008. And wherever we are performing a software validations, we have to follow these current versions for the software validation. So this is the entire history, how the software validations came in existence, especially for the pharmaceutical industry. Now let's talk about the different categories. So I'll just briefing the category comparisons between the GAMP 4 and GAMP 5 guidance, because GAMP 5 is a current version and previous superseded versions, I can say GAMP 4. However, we are not following the GAMP 4 guidance as on today because recent current effective version is 5. Let's understand the different categories based on these regulations. So here it is categorized as a 5 different categories. So if we talk about the category 1, so as per GAMP 4, it is considered as an operating system. But if you see the revised and current effective guidance, it talk about the infrastructure. We will see the details for these all the sections, how it can be how we can interpret these. Second one is category 2. So previously it was dedicated for the firmware, but nowadays it is not longer used. What happens? Some of the firmwares were distributed into the infrastructure, and somewhere is some of them is classified at a non-configurable product, and that's why category 2 is not exist as on today. Now we talk about the category 3. It's a standard software in previous versions. Now it is considered as a non-configurable product. Category 4, configurable in previous versions, again the configurable product. So there is not change in this categorization. For category 5, it is considered, previously it was there custom software, now it is custom applications, because we have to see the broad perspective. Now let's move on to the system. So what do you mean by system? Because in computerized system, first we can able to understand the concept of system. And as per the American Standard of Institute, system is a consist of man, machine, and material. So all together is considered as a system. Anytime you can be considered as a computerized system, it is considered as a laboratory system, it is considered as any manufacturing system, or maybe quality system. So all together, these 3 primary ingredient is there. Now let's talk about the computerized system. So when we talk about the computerized systems, we obviously know that what do you mean by computer and what do you mean by systems. We already understood the concept of systems. Now for the computers, we have to consider as a hardware. We can also consider as a software. Some of the peripheral device, like suppose if you want to attach with the printer and you want to take the printout from the chromatogram. So again, peripheral device is also considered as a computerized system. What happens if you have performed the UV spectrophotometric analysis, and whatever data generated, you can directly store into the network drive or maybe a cloud infra drive. So what happens? These are the backup drive, or we can say server network drive. So this is also considered as a computerized system when we talk about our software validations. And obviously, personal like you and me, all I can say consider, we all are considered as a personals if we are involving in computer system validation aspect. And we know that as a persons of quality assurance, whatever documented evidence, I mean associated documents, that will be considered as a computerized system, and all together is a very crucial and important during the software validation. Now let's move toward the concept of validations. As we know, the validations, it can be applicable for the software validations, method validations, process validations, as we will also see the cleaning validations in I think in next presentation. So we understood what do you mean by the validation. So if you see the guidance, it is talk about the documented evidence. I'm talking about the documented evidence. If you have a, if you don't document it, so it doesn't happen. As a thumb rule for the quality assurance, especially in pharma industry, it must be documented. Without evidence, we cannot prove that. And if you see in court and judge, again, they can take the decision taken based on the documented evidence. So they can also see the evidence, maybe a documented evidence, maybe some photo evidence, maybe some video evidence. So this will also consider as a documented evidence, which provides a high degree of assurance. Again, it is talk about the high degree of assurance. So you have to interpret how it is considered as a high degree, and that system work as per intended use with predetermined compliance. I mean, suppose if you want to develop a pen. So for pen, what is your basic concept? It can be used for the writing, or it can be used for the sketching of pictures like that. So we can know the intended purpose of that particular product. Likewise, for the software, for any product, it should be system. If for any system, it should be work for its intended purpose. So main purpose we have to identify, and accordingly it must work with predetermined compliance. So what do you mean by predetermined compliance? I mean, it can be considered as a quality attribute. So whatever requirement is there, guidance requirement is there, we have to compliance this requirement. So in only validations, you can understand what meaning is mentioned. Documented evidence system work consistently during intended use. Again, it is a consistently. What do you mean by consistently? If you perform the software validations, if you work with these softwares and you can get a result, same results you can get after 5 years, after 10 years. If it is used anytime and it is in live, I mean, we can say life cycle. So we can say it give the consistent results whenever we are using this software. And as I talk about the quality attributes, if every guidance requirements, we have to satisfy this requirement. Now we see the counterpart. What do you mean by the qualifications? Because in software validations, we have to perform the validation activity. But in case of server, or we can say network drive, so there is not a validation, but instead of that, we are performing a qualification. So what do you mean by qualifications? Process of assurance that specific systems, premises, equipments are able to achieve a predetermined acceptance criteria to confirm attributes what is purposed to do. So again, definition wise, there is not a major difference between validations and qualification, but during a validations, we have to provide the list too much extent of validations. But in qualifications, whatever design specifications or whatever calibration criteria or qualification criteria is there, If we meet our network for the qualifications, we can consider as a qualified network drive. Let's move toward the software validations. And as if you see the CGMP, and I think you can aware, what do you mean by CGMP? Current good manufacturing practice. So whenever we use any software in CGMP, I mean in manufacturing process, this software is known as a CGMP software. And for guidance, as per guidance requirement, every software workflow must be validated appropriately. So I mean every software, or we can say each software. So we cannot have a validate some of selective or specific software. Suppose 10 number of software available in your department, you must validate 10. If you are not able to validate 10 software, but instead of that you are able to validate 8 software, and you are not able to validate remaining 2 software, so you have to provide the scientific justifications why you are not able to validate remaining 2 software. Again, it is mentioned at appropriately. So it is up to you how you can define the appropriateness for the software, because each and every individual softwares have a different workflow and different type of functionality. Accordingly, you have to evaluate the appropriateness, and accordingly you have to validate your software. Now we talk about the extent of validation. So it is a more critical, because it is a purely depends on risk, criticality, and complexity of that system. So what do you mean by the risk? Risk, we can say probability of harm is considered as a risk. And if you want to calculate the risk, we have to think about the severity. We have to think the occurrence, or we can say probability of that occurrence. And we can also think about the detectability. So all together we can able to calculate the risk. Again, criticality is considered as how critical your software is during your applications. If you want to use your software for the manufacturing and it is a very critical, so you have to do the validations extent up to that. Again, the complexity, because what happens if you want to use your data logger, I mean data logger temperature monitoring device. So it is a simple device. You have to just device which capture the data, and which data is automatically transferred into the software, into the computer, and you can take a printout and you can use this system. So this is the simple system where the extent of validation is very low. But if you talk about the complex system, I mean you have to prepare some customized software. Especially, suppose I'll just give the example. Saraswati Pharmacy College, Saraswati College develop one software, and it is applicable only for the Saraswati Institute of Pharmaceutical Science. So it is a special designs for the special application for the special students of Saraswati Pharmacy College. And that's why it is considered as a customized software. And for the customized software, depending on your workflow, you have to categorize as a complex product, and that's why the extent of validation is more for that complex product. Now let's understand the scope. So what happens? What is the scope of your software validation? So it must satisfy the scope of application. Suppose I'll just talk about the UV visible spectrophotometer. So what happens if you want to use the UV visible spectrophotometer for the qualitative purpose? I mean, just to identify the lambda max, and you can say that your compound is paracetamol, and you are validated for the qualitative purpose. But in real applications, if you want to use the same software for the assay of paracetamols by using a UV visible spectrophotometry method, so what happens? Scope of application is a quantitative, but during validations, you have to perform a qualitative exercise. So that is not acceptable anywhere. What is the scope? Scope is totally depends on the scope of applications. Now, validating workflow ensure the intended steps, requirements, and calculations in softwares which are accurate and perform properly. And that's a research how it is worked. It should be give the consistent and accurate results. Implement the appropriate controls to manage the risk associated with each element of system. What happens, because each and every vendors, every software have a different vendors, and for the different level of R&D of that particular software, the complexity involved and risk involved in that system may varies from software to software. And that's why we have to able to manage risk based on our applications. Control that appropriately designs to validate system for intended use. As I talk about software, hardware, personals, or document related activities. Now let's talk about the V model. So V model is nothing but the software validations models. It is because the shape you can visualize, it is a clearly shows as V shape. And that's the reason it considered as a V model. So whenever we talk about a validation, so first document for our validation is validation plan. And based on that validation plan, so what do you mean by plan? So how to perform the software validations? I mean, what is the objective? What is the we can say different requirement for your software? You have to define prospectively before starting of validation, and based on that, we have to think about the URS. URS means user requirement specification. So let's understand the URS. Suppose if you want to purchase any mobile, because nowadays we can say mobile generation peoples are there, and what we have some expectations from the mobiles have a capability to have a Wi-Fi capacity. Mobiles give the Android 4 versions, have a storing capacities, how much data can I store, what is the functionality with respect to technical aspect, security aspect. Mobile can capables to book your ticket or maybe your special application. So these are the URS for your mobile. Likewise, for whenever we talk about a software, so what is your expectation from this software? You have to document it, and after that we have to perform the testing. Now we talk about the after URS, it's come under the functional specification. So what do you mean by the functional? So whatever function given in your mobile, is it correctly work or not? And that's why we have functional specification is there. And after that, it's come as a design specification. And finally, design specification is not a part of our side. It's a vendor requirements, vendors responsibility. They can perform the design specifications and they prepare the system build. So system builds means the especially how to software is designed and developed. And that is also considered as one of the critical aspect for the software validations, because based if they not understand the user requirement, so they cannot build the system as per the requirement of the customer. And if software is not as per requirement of customer, they cannot, that means customer cannot buy the software. And that's why URS, functional specification, design specifications, and finally based on all these input, system built is there. After that, we are performing a IQ. IQ is nothing but the installation qualifications. So what do you mean by installation qualifications? You can see that your software is correctly installed in your PC or not. And that's why some of the basic prerequisite for the software is there. Because sometimes what happens, you require this some type of configurations, like the RAM is required, like hard disk, or we can say operating system to support this system. And these all requirements we are testing again the design specification. So in design specification, there is a QC criteria, or we can say if it is meet the criteria, then we can say consider as it is acceptable. If it is not within acceptance criteria, we can say that it is fail in qualifications at a installation level. Once the installation qualification is over, we can think about the operational qualification. So in operation qualifications, we have performed the whatever operations from the software. We can perform the testing against the functional specifications. After that, it will come the performance qualifications. And performance qualification is a requirement from the user side. Performance qualification is a requirement from the user side. So what happens? user can test what they want from the software based on the applications of that software. And finally, all together, it's come into the validations report. So what happens from the plannings to the validation report? This entire process is considered as a software validations. Whatever software is there, and depth of validation may even varies, still we have to follow this workflow for the validation. Let's see the details workflow, how it is come into the picture. So first, as I talk about the software classifications or categorizations, depending on the classifications, we have to perform the vendor assessment, because if your categories, if your software is having a category 3, the vendor assessments you can complete by using a questionnaire aspect. So you cannot go to the vendor site. You just send the questions and answer, and they fill the questions and answer and provide the documented evidence, and that is enough. But if you want to talk about the category 4 or higher, so what happens? You can go there and you can audit these systems and the process followed by the vendor. So that is the on-site audit. And after the clearance from the quality assurance for the vendor assessment, we can think about the URS preparations. As you all people know what do you mean by URS and what is the requirements? Because if we not give the demand, so how we can satisfy the demand? So you have to list down your demand, and then you can talk, you can think about this demand. Then it comes to the validation plan. So again, validation plan is the, as I talked prospective documents. After that, we are preparing a test plan, and test plan preparation is the responsibility of the technical owner, means IT side. After that, we are performing a workstation qualifications and configuration specification. So what do you mean by the workstation qualification? So in workstation qualification, suppose I want to install the software in my PC. So I just want to do some of the prerequisite for this PC. And for this PC, I just want to log the date and time setting. What happens? Suppose if you are not able to system clock, I cannot able to restrict. So what happens? Anybody can change the time. So if time is changed, you can get the variable time in your analysis. And that's why we have to perform some of the workstation qualification aspect. It is not only related to time. There is so many parameters. We have to think about the password policy. We have to think about the user requirement, enforcement requirements. So all requirements, especially for the window applications or operating system, we have to qualify the software workstation first before the starting the validations. And then we can go about then we can start the installation qualifications, operational qualifications, and PQ qualification. So we are preparing a different test cases. I mean, you have to prepare the template, and you can execute the each test, and you can generate the test evidences. And this test evidence is provide the evidence, because what happens whenever auditor came, they just see the test evidence, and based on this test evidence, they assume that software was validated as per 21 CFR Part 11 requirements. After that, we are preparing a test reports. And one more thing here is requirement traceability matrix. So what do you mean by the requirement traceability matrix? Suppose in URS, you have to mention the 100 requirement, and during a testing, you are performing only completed the 80 requirement. So what about a 20 requirement? So you have to link one documents where you have one side there is a URS, and other side you have to IQ, OQ, PQ test cases number. In which particular test case you have to test it, and if it is not tested, is it controlled through the procedural control or any technical control? If it is not justified properly, your validation is not considered as a complete validation. If in URS you have to mention 100 requirements, you have to test 100 requirements during the testing. And finally, we are preparing the validations report, and based on that validations report, we are preparing a system release report. So in validation report, we have to include the IQ, OQ, PQ results, and if something is not as appropriate, because if anything it deviate from the validation plan, so that will be considered as a test report problems, and test report problem is also identified in validation report. And finally, we are release the system based on system release report. And system release report is considered as like if you are completed B Farm and you get the convocation certificate from the university. So you are the system, the convocation certificate is considered as a system release and you are ready for the job. Likewise, for the software, once you validate it, there is a one certificate, we can say system release report and certificate, and based on that, we can use the software for the routine applications. And now we cannot say that it is a consider that it is a up to a validation part. Now, what's about for the in life? I mean, now you want to use the software. So during this usage, there is a system admin procedure. So what do you mean by system admin procedure? So suppose there is a one software, how many users you have to provide give the access for that. What is the password policy? If some user left the organizations, how we can handle this user? What is the roles and privileges for that particular role? Because analyst cannot be able to delete any chromatogram from the system. So likewise, this will be considered as a system admin procedure. We can also consider like a periodic review exercise. So that is also, you have validated the systems today. Now after 6 months or 1 year, you have to revalidate, or you can ensure the validity of your software. And more about, I can talk about the BCP, business continuity plan. So what do you mean by the BCP? BCP is required when your software is having a malfunctioning and you are not able to work on that software. So what is any alternative approach to continue your business? And that's why it is considered as a business continuity plan. Now, in contrast to that, there is a disaster recovery plan. So what do you mean by disaster recovery plan? Now you have a business continuity plans to go ahead how it can be. But what happens in your software? Is there any malfunctioning? How we can correct this malfunctioning and how we can rework the systems and come into the live. So that part is considered as a disaster recovery plan. And we have to define the procedure for the business continuity plan and disaster recovery plan. Let me talk about the software classification. So as I told, complexity and risk based on these two parameters. Software is and one more parameter I can intended use. The software is categorized. And as I say, the GAMP5, there are 4 different categories: category 1, category 3, category 4, and category 5. So let's talk about the category 1. So in category 1, infrastructure software, for example, like operating systems, Windows or like that, or maybe database manager. So that will be considered as a category 1 software. So it can be considered as an infrastructure software. And for that infrastructure software, as per GAMP5, we have to perform the qualifications. Validation is not required, especially for category 1 software. Now let's talk about the category 3. So category 3 is we can say non-configurable software. So what do you mean by the non-configurable softwares? I'm just say that commercially off the shelf COT software, or we can say laboratory instrument. Suppose UV visible spectrophotometer is there, IR is there, NMR is there. So whatever inbuilt software is there, we are not able to change the configuration settings. We have to use as it is. Whatever functionality provided by vendor, same functionality we are using for our application. So that type of software is known as a non-configurable software. And accordingly, we have to perform the limited validations for that software. Now let's talk about the category 4. So what happens sometimes in that particular software, you have a ability to configure your workflow based on your requirement. So if you want to configure your product like a LIMS, laboratory information management systems, document control systems, chromatographic data management software. So these are considered as a configured product, and depending on workflow, you have to change the workflow, and that's why you have to perform the more validation compared to category 3. Now let's talk about the category 5. Again, as I talk about the customized software, Saraswati College of Pharmacy specially designed, and they use only the Saraswati College students. So that is in-house software. And for that particular applications, for that particular workflow, you have to provide the extensive validations, because it is not available of the commercial. It is not available at a commercial scale. It is only limited for the R&D purpose or at the one particular locations. That's why we have to perform the depth validations for the category 5. So as I go from the category 1 to category 5, the complexity in software is increased. Likewise, risk is also associated risk with the software is also increased from category 1 to category 5. So for category 5, high risk product is there. So we have to perform the more stringent validations compared to category 3. Now let's talk about the category 3 softwares. So it's a non-configurable product. Again, we have to first documents, we can say URS, user requirement specifications, and you have to prepare means vendor prepare the non-configurable product, requirement testing, IQPQ, and verified against the URS. So what are the deliverables from the category 3? You have a prepared URS, vendor assessment, software categorization. Again, software categorization, we have to ask some of the questions. Is your software used for manufacturing purpose? Is your software used for the control in laboratory control? I mean, HPLC instrument where you have to use the software. If you want to use your software for the BAB study, you want to use the software for the quality management elements handling. So these are the different questions you can categorize that your software is considered as CXP or not. You have to categorize based on the category 1 to 5. You can also categorize based on the electronic record and electronic signature requirement and business continuity. So altogether, you can categorize the software, and accordingly, we have to give the validation efforts. Now let's talk about the audit trails. Audit trail is a very, very crucial and important aspect, especially in pharmaceutical industries. So what is audit trail? Whenever you generate any data, system have a capability to capture the data. Suppose I just acquire the run. I just log in the system, put the password, and prepare the batch and submit the batch and save the result, print the results. So all type of activity is automatically captured based on username, date and time, what they do, why they do, all information, all 5 questions is satisfied by this audit trail. And nobody have a rights to disable or enable the audit trail. Only admin can make the change in this particular audit trail setting. If user are able to disable the audit trail, so you cannot get the information, is it really perform as per regulations or not. And that's why whenever any audit is there, they ask about the audit trail. Show me the audit trail. What is captured? Anything deleted from the audit trail? So whenever you delete any chromatogram or any samples from the batch, it is automatically captured into the audit trail. And that's a reason audit trail review is a very critical. And that's why the setting of audit trail is not under control of laboratory persons or lab manager. It is on control or independence control with the IT team. And most about data integrity, as you see, I think third lecture, it's talk about data integrity. You can able to understand the ALCOA plus principle. So I'm not going too much details about data integrity. And finally, I'll talk just about the SOPs, standard operating procedure. So what do you mean by the SOP? It's a document which provide the instruction how to perform, how to operate, and how to execute your test. So it is a detailed step-by-step procedure. It is mentioned in standard operating procedure, but it is a prospective document. You can prepare the SOP and then you can start your work. And for analysis, we are considered as a method SOP, or we can say ATP, analytical test procedure. So this is also required for the any activity. So in our software, how to operate the software, we have to also prepare the standard operating procedure. Let's talk about the category 4, configurable product. So as you see here in category 3 requirements, you can have a list requirement. But if you see in category 4, user requirements, functional requirements, again, additionally, we can say configurable configuration specifications and configure product, configuration testing, functional testing, and requirement testing. So you have to perform all this testing for the configurable product. So what are the deliverables? As I talk about the same deliverable, additional deliverable is the functional risk assessment, because here functional specification is there. And what is the functional testing? And based on that testing, you can identify the risk, and you have to define or control or we can define the mitigation strategy for that particular risk. So that is the additional parameter, especially for the category 4 document for software. Now let's talk about a category 5. So as I say, it's a customized, or we can say custom product or custom software. So again, depth of validation is too much high. Here it is a design specification and module specifications, and you can go for the system build, module testing, and design testing. And finally, if you see the deliverable, so here the deliverables are, if you see, there is a 3 additional deliverable like design specification, functional risk assessment, and we can say functional testing. So these are the additional compared to the category 3 software. Let me see here, just give the difference. Here in category 3, we have to talk about system requirement and risk assessment. For category 2, category 4, we are additionally perform the design specifications and operational qualification. And for category 5, with respect to category 4, only the build review is an additional document. Now let's talk about the responsibility, because without responsibility, how we can say that software validation is completed? Because man, material, and method. So man is also critical role, and we can say for the software validation. So here there is a 4 major stakeholders are involved. First one, I talk about the business owners. So business owners to fulfill the business requirement. So suppose I'm an analyst and I'm handling the HPLC instrument. So what I expect from this HPLC software? So that requirement is provided by the business owner. Now let's talk about the technical owner. So again, it is a provide the IT related technical supports during the life cycle. So what do you mean by life cycle? So from the software creation, from software validations, implementation, uses, until you have to use at the destruction level. So these are considered as a life cycle of your software. And this support is provided by the technical owner. So whatever administrative related activity, which are not under control of laboratory, that is taken care by the technical owner. Now we talk about validation lead. So validations leads to provide and approve the validation related documents. Because if you see the entire workflow, we have near to 20 documents. So all documents must be reviewed by the validation lead. And finally, I'll talk about the quality assurance. So what is the job here is to review and approve the documents against the quality requirements, validation requirement, and additionally the compliance requirement. So what is stated in guidance and what is stated in your SOP or what is stated in your validation plan, it should be a mimic. Otherwise, there is a gap between the guidance requirement and in your SOP. So that is not acceptable. And that is the job of quality assurance during the software validation. Now let's talk about the other important aspect. Now I'll take only 3 minutes to complete the rest of part. So other important aspects. So whenever we talk about new softwares or upgradations of software or operating systems or any retirement, there is a change control management system is there. So what do you mean by the change control? So if you want to change from the existing software list or any software inventory, you have to raise the change control, and accordingly you have to handle this change, and you have to give the proper justifications why this change is required, and what are the different action items for that particular change, what is the timelines, who are the reviews the change control, who are the approve, and who are create, review and approve the change control from the department side, and may if any review from the quality, what is the responsibility from quality. Now for the periodic review, as I talk about the user access. So in user access, it is a must requirement that only authorized user can access the software, and unauthorized user cannot access the software. So it is a clearly mentioned. So that means you have a list of authorized user. You have to give the permissions to use the software. So there is a you have to documented evidence that that much users have this particular role. Now we talk about the privileges verification. So what do you mean by the privilege verifications? Because what happens, there is a role, is the role based software is there. I mean, suppose analyst role, quality assurance review role, or administrator role, or manager role. So every roles have every different privileges. Analyst cannot be access the administrator related activity, otherwise they make the change in this software, they can make the deletions from the software. That's why privileges verifications and role based access is a mandatory requirement for the any software. Now let's talk about every day you are creating a new records, or you are performing a new analysis or any data in software. So there is a backup verification process is there, and you have a backup retrieval process already available, should be available for that particular software. Group policy verifications, because what happens, group login means general login, like a user, and you just put a password, user and password, it is not acceptable. User can consider as a Haresh, user can consider anybody from any student, so that you cannot have a traceability to attribute that functions who perform what activity. And that's why user name must be displayed, or user code must be have a access in your software, and that username is also captured in your audit trail. So anybody can see that what activity performed by what particular user. Let's talk about the migrations. Again, migration, it is required when we upgrade the software from the version 1 to version 2. So you can see the compatibility of data of previous versions in new versions. You can see the completeness, how the complete data is transferred into the backup, and how complete data is transferred in existing system. So that are known as migrations of data. And finally, I can say about the revalidations. Suppose if you upgrade your software, if you upgrade your operating system, so what happens? You reload the software, and if you reload the software or reinstall the software, again revalidations come in picture. And depending on the change, depending on the aptitude of change, we have to perform the revalidation experiment. Now let's see the examples here. I'll just talk about the analyst software. So it's a non-configurable product standard, whatever software given by vendor, same software we are using. So in URS, what we can do, we are having the 21 CFR Part 11 requirement, electronic record, electronic signature standards. What is the expectation from software? Because this software is specially for the LCMS instrument. So in LCMS instrument, I just want to tune compound, because I want to identify the molecular weight of your compound. Sample preparations, acquisition method, quantification method, acquiring of sample, batch integrations, batch saving, printout, chromatograms, and finally the backup in archival procedure. So these are the user requirements specially for this analyst software. And depending on the type of applications and usage, your URS may varies, and accordingly we are performing the software validation. Now in summarized, we taught about the software validations for its intended purpose. We already discussed about the vendor assessment, how it is applicable for each application. We also discuss about the documented evidences required for these to support the software validation. Again, Validation documents depends on the software classification. Each software must be validated. You can see the must requirement each software. So nobody can exclude. There is no exclusion criteria, no exceptional criteria. Whatever software available in laboratory, you have to validate it. You must have to validate it. Each infra component must be qualified. Software must comply the guidance requirements. System shall use after the completion of validation. I mean, without validations, we cannot use the system for the routine use. System admin plan, business continuity plan, disaster recovery plan shall be prepared to handle the day-to-day activity. Extensive validations must require for the category 5 as we talk, because category 5 is a customized software, and for that customized software, we have to provide the extensive validation. And with this word, I will end up my presentations. Yes, if you have any questions, you can ready to ask.
If you have any questions for Dr. Harish, please add them in chat box, and we will be able to answer some of them during the question answer session.
Yeah, sir, one question is there, that which are the important software currently used for the validation in the industry?
Wow. It's a very nice question. Especially if you want to talk about the software. Suppose I am working in production environment. So whatever software related to productions, because in productions there is some specific instrument specific software is there. Some of the software may be used for like a laboratory, I mean, environment control software is there. Especially I'm talking about if you want to maintain the room temperature and percentage relative humidity, how it is captured and is it within acceptance criteria or not. Because each and every rooms have some specific temperature and humidity limit. And that's why that type of environment control software is also used for the production. So what type of software you are used? Any software which are directly inbuilt in your PC, and it is used for the any GXP related. I am talking about a GXP, good clinical practice, good laboratory practice, good manufacturing practice, any type of activity, documentation practice, we have to validate. Now, apart from this production environment, if you talk about R&D environment, so again in R&D, if persons are working in quality assurance department, so they are using like a quality management system software. I mean, track-wise software, which are able to handle the change control, deviation, kappa. So these are the different elements which are maintained and handled through the quality management system. So for that, they have to validate that particular workflow. Now, if you talk about the laboratory perspective, maybe analytical lab or maybe a QC lab, so they have like a HPLC instrument, and that HPLC instrument is connected with, I mean, it's a maybe any software, any PC, because whatever data generated, it's directly come into the PC and directly we can take for the backup and we can so this data for the future reference. So these are the requirements. Likewise, if you are working in formulations, organic synthesis, or any other departments, whatever software is there, we must validate if it is fall under GXP category. Otherwise, if it is not GXP category, you can waive up. However, if you see the server related, as I talk about operating system server related, these are not considered as a software, it is considered as an infrastructure component. And in that particular case, we are considered as a qualification criteria rather than validation criteria. Hopefully, I can explain your doubt. If any further clarification, I'll be ready to answer further.
Thank you very much, sir. Sir, you have shared the knowledge and information about the computer system validation, classification of software, regulatory requirements of computer system validation, and uh, the revalidation requirements as well. Sir, this information will be definitely helpful to all the participants who are currently working or who want to go in this area. So your session is very much informative, and thank you so much, sir, for sharing your knowledge with us.
Okay, thank you so much, Charmi. If anybody have any questions, you can collect the questions. I will be, and you can sum up the questions and send to me. So I'll be happy to answer your questions if any questions are pending from my side. And in future, if any questions related to computer system validations, I'll be happy to answer that. And with this word, I'll thanks to Dr. Navneet sir and Dr. Ankit sir for providing me this such a good opportunity to deliver such a wonderful sessions. And if you see the participants number is 304. I mean, at the time of initiations, it was near to 210. Now the toll is increase more than, I mean, more than 33% is increase is there. So it's a nice audience, and thank you so much for your patience and listening me. Thank you so much.
Thank you, sir.
Proceeding further, I would like to introduce our second speaker, Mr. Yogesh Mandalia, sir. Sir has more than 10 years of industrial experience. Uh, he has completed his MPharm from from our institute, Saraswati Institute of Pharmaceutical Sciences. So he is alumni of our institute. Next, sir has completed his MPharm in quality assurance. Now, sir has joined the industry as trainee in Alembic Pharma. After that, he was associated with Zydus Cadila as team lead, QMS and validation. Currently, he is working as manager, audit and compliance cell at Sun Pharma. Sir has expertise in quality system management and manufacturing, and sir has leading role and responsibilities in investigation of manufacturing and analytical system, QRM and MQRM. Sir has proficiency in handling the audits like FDA, MHRA and various national audits also. Sir has conducted the trainings and also has taken some trainings for GMP, GLP, etc. Now I would like to request Yogesh Mandalia sir to deliver the talk on his topic, that is cleaning validation.
Yeah.
Please, sir.
So thank you very much, Charmi, for good introductions. So first I will thank for Ankit Chaudhary sir, Bhumi Patel madam, and this is a very nice arrangement for this webinar. So we will start with the cleaning validation approach. Before we start the cleaning validation activity, why cleaning validation is important in the industry? You can also imagine in the house also that replications, the same industry following for the cleaning validation, the same following in the house. So that in the cleaning validation program, before I starting, I just give one example to you. So that you can understand what exactly approach for the cleaning validation and why cleaning validation is so much important in the industry. Because is now decade, you can review the US agencies, MHR agencies. They are the many of the 483s, many are the observation on the cleaning validations, on the cleaning. So that I give the one example, then we will start the basic approach on the cleaning validations. So you know about all of the, once you cut some garlics or onion with the knife. And the same knife is used for the fruit cuttings. You can identify that some of the smells, some of the traces from the previous onion or garlic, it carry over to the next product, means next product. You clean the equipment, immediately you identify that the previous product traces in the food. So that it is identifiable, and you can identify that, yes, this is a smell for the garlic, this is a smell for the onions. But in the industry, you manufacture the paracetamol and then you next for the methotrexate, you cannot identify easily the paracetamol traces carry over on the methotrexate product. So how can you justify or how can you allocate or how can you demonstrate this is the product is clean and that is not carry over from the previous product? So that you should be demonstration of the practically cleaning validation in your all equipment, product, and you can prove that scientifically my product cleaning procedure is robust. So this is the concept in the cleaning validation what we use and why cleaning validation is so.
Yogesh sir, sorry for interruption, but your voice is breaking somewhat.
Yeah.
Is there any network issue? Let's see.
Okay, I will check. So now is clearly audible?
Yes, sir.
Yeah, okay. So we will discuss clinical validation is a very vast subject and we cannot cover in this 1 hour sessions, but I will be varnish of your clinical validation concept and what is the exactly clinical validations. It will be defined and basic approach why clinical validation is most important part in the industries. So during this sessions, we cover the historical of the background for the clinical validations, different type of cleaning method which are being used in the industry, what type of cleaning use, worst case, how can identify the worst case, what is the matrix approach used, evaluation of the cleaning procedures, current regulatory requirement. So these are the concepts will be discussed in the presentation. So the cleaning validation first approach is introduced in the industry in 1984 and subsequently the 1988, 1992, 1998, 2005, 2015 and 2020. So now 1982, 2022, why these many guidelines and many publications has revised this approach and continue to develop the cleaning validation because they are the still open subject to discussion. So just I will discuss about the major cross-contamination event in the occur in the world. So that one of the event which increased in the FDA awareness of the potential for the cross-contamination due to inadequate procedure was in the 1988 record for the finished drug product in the cholesterol resins. The bulk pharmaceutical and chemical used to the product had become the contaminated with low level of the intermediate and detergent from the production and agriculture pesticide. So what exactly happened in this case? They have the usage of the previous product drum used in the cholesterol product manufacturing. So previous product traces carry over in the next product for the cholesterol and they have identified the serious impurities, serious side effects and occurrence. This can be highlighted to the FDA. That's the cleaning validation approach and the cross-contamination approach introduced in the industries. So currently, subsequently that 1988, 2000, 2005, 2015, 2018 and 2020. So currently we are using for the HBEL approach for the health-based exposure limit. How can we carry over? How can we identify what method calculation? This concept, this word, I will be discussing in this presentation. Do you aware about of that? What is the HBL? What is the LD? What is the PDE? This will be discussed. So you know all about of that cleaning validation definitions. I cannot read this cleaning validation definition, but I will just simplify that cleaning validation definition. So you bifurcate two words, cleaning and validations. What validations? Validation means you have a data, you have a predefined, predetermined result. You just matching with the acceptance limit. It's called as a validation. What is the cleaning? So cleaning, whatever you, any of the equipment clean, you have a procedure for the cleaning. You should be your product no carryover from the previous to next product. Your microorganism is free. So this type of cleaning part will be carried over, and that contributing to the validation. The process providing the high degree assurance will develop the method free from carryover, free from microorganism, and usage of cleaning agent which can be helped to validate your predetermined result. It's called as a cleaning validation. In industry, what type of cleaning method usage? There are the three type of cleaning method are using. First is a clean in place. So you just out there. So what is the clean in place? So the clean in place means automatic system. They are the recipient system. You just start the button, start the cleaning, then first potable water cleaning, then it's purified water cleaning, and then completely dry the equipment. You need not intervention during the cleaning procedure. That's called as a CIP. Most of the large industries, our large facility, our most of the industry using and adopted this CIP method. Apart from that, the second method is the COP method. So COP method is the some of the equipment, some of the parts you dismantle and then you clean in the another equipment. Like I will give the one example, like the bean washing machine. So what you manufacturing the product, you bean blender, you perform the activity, then you dismantle the bean and transfer to the bean washing machine. So then your cleaning done to the bean washing machine. So that you identify the some of the dismantle parts, some of the automatic parts, these are the combination. It's called as the COP method. And the third one is the manual cleaning method. It is the like the mobile equipment, accessories, this type of equipment, intervention of the manual cleaning, that's called as the manual cleaning method. So industry, manual cleaning method is avoidable and they try to best they can avoid this method. Why? Because some healthy person then can be pressurized 10 pressures. Some of the unhealthy person they pressurized during the cleaning 5 mm pressure. So these type of pressure or scrubbing, these type of the different variable parameter which can be contributed that your cleaning is not adequate or properly, and there is chance to previous product traces carry over to the next product. Next, we'll discuss on the you listen about the type B cleanings, type A cleaning, general cleaning. So what is this funda for the type B cleanings? I just give them an example. Type B cleaning means product changeovers. Like you clean the paracetamol, you are manufacturing the paracetamol product, and then you clean the product with the weight cleaning, and then you start the next product of the olenjapine. So this the between the product you perform the cleaning. It's called as a weight cleaning. And give the example for the type A cleaning. You perform the paracetamol product manufacturing and the same product manufacturing of the paracetamol. So you do not need the weight cleaning required. You generate the data, if scientific rationale established, based on this document evidence, you can identify and you can consider this is my type A cleaning from this product to this product. How many batches? This one of the word using in the industry as a campaign cleaning. Like you can manufacturing 10 product of the paracetamol between the batches, you can clean only the dry cleaning. You do not require for the wet cleaning. I give the one example in the social life. How can you contribute the type A cleaning and type B cleaning? In the morning, you wake up and your mother will be make for your roti. So first you eat the roti and you go to the office.
Yogesh sir, sorry to interrupt you, but can you please change to the slide mode, your presentation?
Yeah. Yeah.
Thank you, sir.
Yeah. So your morning you take the some rotis and you go to the office. And second, your child is wake up and your mother will make a roti with the same rollers and same the roti maker. So that time your mother will be only just dry cleaning on the roti maker and your roller. So this is the procedure considering as a type A cleaning. For apart from that type B cleaning, you just take the some food on the night and you clean your equipment and you keep on the place. And the second day you another food make for this process. Before you start the process, you can check the your equipment, accessory, and clean our map. So this is the cleaning, the procedure. It's as a type B cleaning. So in the industrial approach, these are the type B cleaning and type A cleaning. How contributed in the cleaning validations, this will be based on the health-based approach, acceptable daily dose limit, and permittable daily exposure. So this contribution will be discussed in the next slides with the details, what is the health-based approach and what is the ADE and PDE. We'll discuss on the next slide. The same will be discussed on the dose approach and the 10 ppm approach, how we follow and what industrial criteria for the cleaning validations. In this discussion, before we go into that, we just understood the what is the MECO. If we go into the some equipment clean and take the swab sample and send to the quality control for the analysis, that time you define some limits. For this X product, my limit is this. So in cleaning validations, the name of this word is called as a MECO, maximum allowable carryover value. So this is called as a MECO calculation, and this word will be used in the next slides for your understanding. So that just I updated to you. Now we understand the what basic philosophy of the cleaning validations. Now we going towards for the what exactly approach used in the industries. So you just considering you have a new equipment introduced in the facility. So what you criteria follow? First, you raise the change control. Whatever in industry came for the equipment, product, you first raise the change control. I have procured this material, I have procured this equipment, I have introduced this product in the facility through this change control. So that the detail and the integral part will be introduced in the change control program. And you can define the impact assessment. What is the basic requirement to complete this process or establish the equipment in the industry? So first you cut the equipment, you're developing and checking the cleaning procedure. You take the example for the compression machine. You just receive the compression machine. You just check the cleaning procedure. Have you existing cleaning procedure established or not? If not, then you can take from the vendor or you try to the some established criteria based on your scale-up batches so that you develop the cleaning procedure. Based on this cleaning procedure, you can what criteria define? What is the surface area? Like the compression machine total surface area. What is the design? What is the capacity? What is the operating principle? This should be considered. Based on this considering, you can establish your procedure. You can establish this procedure. How can you identify that? On which equipment, what location I will take the swab sample. So that is the major questions. How can I identify that this is my swab locations? How can I identify that you can confirm that the scientifically this location is my hard to clean and there is no need to change the location during the entire cleaning process. So that what industry is following that you perform the accessible location. You identify that my compression machine is a 10 location, you can identify accessible location. Means you can perform the 12, 15, 16, 20, 25, major, whatever contact surface, you can identify that. You mention in the rationale, this is my accessible location, hard to clean, hard to reach. Based on the operator and supervisable capability, you can identify these locations, 15 location, 20, 25 locations. Then you can perform the cleaning procedure. Whatever in the industry, you just started the cleaning with the potable water. This is called the raw water. Then you take the swab sample from these locations and then same equipment clean with the detergent. Or you can use the solution like the Hamtop solution is most of the industry use. That efficacy study to be performed and established that the product and this equipment is clean with this Hamtop solutions. Then you perform that the potable water. You perform the purified water. So that you first you take the swab sample from the potable water, then you take the detergent cleaning, our solution cleaning, and then you perform the purified water runs. Then you again take the same location swab samples and perform the swab sample. You send to the quality control for the analysis. During this analysis, some of the limits, some of the traces identified in the your potable water and purified water. Because the purified water you can identify wide limit, BQL, no quantification limits. This type of data you can write. But in potable water, you identify that some of the limits or some of the area it is the critical to clean or hard to clean. So that these locations you can considering for your routine commercial or as a product establishment identification hard to clean location, hard to reach locations. This called as a basic funda for identification of hard to clean location and hard to reach locations. The same funda is using for the new product introductions. What in the new product introductions we considering for the AD value, what the AD values, what the daily dose, what is the base size, what is the solubility, what is the hard to clean criteria. If the AD and PD value less than 10 microgram per day, you should be considered this product as a worst case product. The EMA guideline has literature published with the detail of the AD and PD. The basic concept, if your AD and the PD value is less than 10 microgram, you mandatory perform the PD evaluation for the overall product, which have the lowest PD value. If your PD value is more than 10 microgram, you can categorize based on the worst case study and you can perform the cleaning evaluations. So just to be discuss about all this point in detail, what is the criteria? How can I identify? The first criteria, it is the product with the lowest PD. Like we have just discussed about of the, if I have a paracetamol product and we identify during introduction of the product, my AD and PD value is 50 microgram per day. And my next product coming, it would be the 14 microgram per day. So what my worst case considering as per approach 1, it should be my 14 microgram AD/PD value. This should be considered as my worst case. So subsequently you edit the product and you check and verify that my AD/PD value. If my established cleaning procedure 14 microgram per day AD/PD and the equipment is clean and found satisfactory with no carryover, no free from microorganism, that's all. My 14 microgram per day procedure for the equipment and product is established. So above the PD and PD value 50, 60, 70, 80, 90, it is clean 100% because the established procedure is 14 is clean, then above the 14 is 100% clean. Another approach, EMI specifically guides that the 10 microgram, less than 10 microgram, you mandatory perform the cleaning validation at, and also you should consider as a cleaning validation. The third approach is the cleanability. You generate the one discussion points with your operator supervisor who directly relation with the products or directly relation with the cleaning. So what that? He feels that the cleaning is hard to clean, or they clean that it is easily clean, it is a moderate clean. You some in your study you can just throw out the cleanability. It is easy to clean. It is directly you clean the water and you completely identify that this is perfectly okay. But you just go into the approach number 2, that product with the least solubility in the water content. So based on the solubility, what is the API solubility? Highly soluble, springly soluble, easy soluble. So that's solubility. Based on that, you can identify that your worst case. So these 4 criteria and these 4 criteria you use for the cleaning validation for the worst case. I just give you one example for the cleanability. You routinely you use for the some make of food for the your vegetables and some routine food. And someday you prepare some the oily or butter foods, so that what you existing procedure, it is not completely okay to clean this the equipment. you need some additional control, like the usage of the detergent, or you boil the water and then rinse the material and then you clean. So similar approach here is the follow. If you require the cleanability, easy clean, that is perfectly okay. If you find that some of the hard clean or your solubility in insoluble product, so that you require for the some of the materials. I give the some example. You have a listen some of the Utragit. So what is this Utragit? It is the material of the coating materials like. So that Utragit cannot clean easily in the water. So that also this Utragit cannot be cleaned easily to the hot water. That specifically you require for the some of solution. Like the KOH, Utragit can be easily soluble in the KOH solution. So you have establish a procedure with respect to cleanability and solubility. So when you complete your cleaning procedure, then you use the KOH solution and then you confirm that the my product is clean. Then how can you assure that my KOH material is not carried over to the next product? That you can verify in the pH and you confirm that the my cleaning is perfect and KOH is not carrying over to the next product. For the product grouping, just we discuss about the ADE and PDE, like the product list, solubility, like the lowest ADE, PDE, what is the lethal dose, lowest number of doses, what is the API and the excipient solubilities. The same equipment we for the grouping for the tablet manufacturing, capsule manufacturing, because for the tablet, you perform the granulation. In the granulation, you can perform for the FBD, like the fluid bed dryers, fluid bed extruder, and then you compression, then you go in the coating and the inspections and the packing. Apart from that, the capsule filling, the equipment trend is a different. So that you, the based on the product, you can considering the grouping of the equipment. So these are the basic what industry is following that what cleaning procedures. Apart from that, how you take the samples, you perform that your cleaning procedure is established, your macro calculation is perfectly calculated. You identify that this product is my worst case. Now you how to going for the sub-samples. First, you verify the visual inspection. How you visually check for the equipment is properly clean and okay. So that before you go in for the visual inspection, you should be or you can qualify in the inspector qualification program. That you can qualify and scientifically you can say that I have a visibly okay for this process. So that your ophthalmologist, you verify your eyes and confirm it, your near vision and far vision is the okay. Also that you procedure and your SOPs are trained. This procedure is completed, then you perform the visual inspections. In the visual inspection, you can perform the what is the angle, what is the distance, what is the lighting, and you can perform on the lux level. In the whatever you performing in the inspection process, it is mandatory to do some of the lux level is mandatory. It is what that the your light intensity. It is some of the 1500, 2000. That is the mandatory part. For that, your intensity and your eyes can be easily visually this the trashes on the equipment or your material trashes residue. You can verify that. So this is the visual inspections acceptance criteria. Apart from that, you can criteria detection limit like the 1 to 4 microgram per centimeter cube. You can calculate it and the demonstration in your documents. Next, we using for the two swab sampling methods and another for the rinse swab sampling. Now, question is where uses for the swab sampling method and where uses for the rinse sampling. In this case, wherever you have easily identification, the template, like the some of the area, you can identify the hard to clean locations or hard to access it. You can easily identify that we have our 4 by 4, our 10 by 10 centimeter cube. You can take the swab sample. You can easily take the swab sample from surface. But some of the fuse, some of the rollers, some of the brushes, you cannot take the swab sample from the swab pigs so that you can use it for the vein swab sample. We will discuss about what type of swab sample we take. First swab sample we perform for the API. Second, we take the detergent swab sample. What the detergent is carry over for the next product. API means your chemical part you carry over for the next products. And the third part is the microbiological biology. So, what the industry is so what sequence you follow for the swab sampling? You can take the first API swab sampling, or you can take the microbiological bioevidence sample first. So there is a rationale. What behind exactly? Whatever you perform the swab sampling, you mandatory perform the microbiological swab sample first. Because once you take the swab sample, you clean the surface and then you going for the API sampling. So that what exactly happens, the microbiological, the traces, it can be easily cleaned because
Hello, Yogesh sir. I think there is some network issue.